The potential and limits of termites, Isoptera, as decomposers of waste paper products. Journal of Economic Entomology. Date of publication, February 2011. Rationale problem. Termites are indeed capable of attacking a wide range of cellulose-based materials and can inflict damage to other materials without any food value to them, such as plastics, foam board, bitumen, and others, as countless references and reports attest. However, if termites are to be effective decomposers and the intention is to harvest termite biomass, they not only have to attack the chosen food substrate but also to survive and thrive on it. Objectives of the study. It is still questionable whether paper products, with the exception of various filter paper that are commonly used as a food and water holder substrates for laboratory experiments with termites, Molden and Rich 1975, which do not form a significant part of the paper recycling stream, represent a long-term medium for rearing these insects. Essentially, what impact do these substrates have on termite survival? Related literature. Alternatively, termites have been proposed as direct agents for bioconversion of lignocellulosic waste. French 1988 and Toninger 1997, Haritus 1992, Haritus et al. 1993, 1994, Miles 1995, Toninger 1997. In contrast to using enzymes from some of their gut microbes, Okuma 2003, Brown 2007. Because the natural termite diet is cellulose-based, the more paper pulp is recycled may pose additional restrictions on the suitability of a waste paper product as a termite diet. Another related literature, the connections of the quality of the waste products with termite survival and reproduction have rarely been considered in proposals to use termites as decomposers and for biomass reproduction. Toninger 1997, Itakura et al. 2006. Research methods. Termite C. Formasanus groups were obtained from two colonies A and B held in a climate-controlled room, 28 2C and 80% RH, at the Research Institute for Sustainable Humanosphere, Irish, Kyoto University in Japan, Kubota et al. 2008. They were collected from feeding sites within their holding tanks. Ars asperatus was collected from two locations. Colony 1 inhabited pine, Arpinus sp, logs lying on the ground in a public park in Oji, Kyoto, Japan. Both colonies differed in the number of nymphs and larvae in Table 1. Paper types. 10 types of paper were investigated with R. asperatus and only a selection of them with C. Only a limited number of workers was available in Table 2. Several types of quality printing paper, recycled office paper, paper pulp, tissues, newsprint, glossy advertising material, and corrugated cardboard were included in Table 2. Experimental units. Plastic jars, well-vented lids, are 5.8 cm in height, diameter at the base 9.5 cm, 10 cm at the top, were filled with grade 3 vermiculate, or 15 grams, sourced from Australia Exfoliators Pty Ltd, Denum, Australia. According to Widget and Coleman 1970, Cartel et al. 1972, Redford and Doria 1984, Pauletti et al. 2003, Itakura et al. 2006, termites are also of notable nutritional value due to their protein and lipid content, including essential amino and fatty acids. Although higher amounts of some biochemicals, notably lipids, are specific to certain castes such as nymphs and alates, Laughlage, and 1978. Lipid analysis. The preservative or 70% aqueous ethanol used for transporting each sample from Japan to Australia was evaporated under a nitrogen stream, and the termites were dried in a vacuum desiccator at 5 pa over silica gel. Data analysis. Data on consumption. Final termite numbers and weights of each paper were subjected to analysis of variance, or ANOVA, and means were separated with two keys, honestly significant difference, HSD test, P 0.05. Data and percentage were first transformed into arcsine values before ANOVA. Results and discussions. The study was conducted with termites from only 2 colonies of each species. Furthermore, the group of C. Formosanus were exposed to the experimental materials for only 4 weeks, whereas the response of R. asperatus was measured for 8 weeks. C. Formosanus. Paper consumption. Groups feed on all paper types offered to them in Table 3. Consumption, or 0.279 grams, by both colonies of coated top quality print was significantly lower, or F colony A 284.55 DF7, P 0.05, F colony B 20.21 DF4, P 0.05 than of other papers are asperatus. Paper consumption. Groups from both colonies of R. asperatus fed on all papers types and responded in a similar way except for newsprint and Japanese cedar in Table 7. In this experiment, R. asperatus groups were kept at 28 Celsius above the more commonly used 25 Celsius for longer term. Maintenance of groups of this species, example Matsura and Nishida 2001. Lipid content. Only the results for R. asperatus are provided in Table 9. The short experimental period of 4 weekends C. Formosanus and the limited number of paper type and colony combinations under trial did not allow too many conclusions to be drawn. Gas composition. At the start of the experiment, all pre-soldiers and neotenics were removed from groups. This allowed assessment whether or not the type of diet might influence success in forming new soldiers and replacement reproductives in Table 10. Soldiers. Results and discussions. The natural soldier proportion in mature colonies of R. asperatus is 2.5%, according to Matsura 2002. With a relatively small initial group of size of on average 340 termites, only a few soldiers could be expected in the group after 8 weeks. Production of nymph soldiers. Neotenics and reproduction. Species of reticulotermes and groups as isolated from their colony readily from neotenics and commence breeding. Example, reestablishing as a colony. According to Turn 1998, Lane and Wright 2003, Miyata et al. 2004, and Lance 2006. Neotenics can develop from workers or nymphs, although nymph-derived neotenics are more common in R. asperatus, Miyata et al. 2004. Termites as decomposers of paper products. The results indicated that several types of paper could be suitable for breeding. However, negative impacts on termites of several of the paper diets, notably those that compose a bulk of paper waste such as cardboard, paper diets, notably those that compose the bulk of paper waste such as cardboard, newsprint, and magazines. Note 2002 were observed. Conclusion. Waste paper comprises diverse products. In this study, termites fed on the full range of materials. In the longer term, only a few of such products such as scrap pulp and tissue paper, prove suitable as a staple diet of termite cultures and to termite breeding. Although paper consumption traits from several weeks of laboratory trials may encourage the idea that termite could be potential major decomposers of a wide range of waste products. Survival rates, physiological condition, example lipid content, and the ability of termites to reproduce on such a diet are the critical aspects on which the suitability of paper type for decomposition and for termite breeding has to be based. The book of waste paper is made up of cardboard, newsprint, and magazines. And our experiments with our asparagus termites did not fare very well on these three products despite consuming cardboard in much larger amounts than other type of paper. It is quite interesting to study about termite. Termite as decomposer of waste paper products, it will help to reduce the waste. The paper is very relevant because it includes the efficacy of having a prevention economic, especially in biomass. The impact of this paper will help the future researchers as a guide. The methods and data collection is very reliable, and I agreed on this. However, termites is continuously spreads and affects many houses, which destroys the furniture and woods. That is why they conduct this study in order the termites can do goods as decomposers. As the researchers made the right conclusion, and it's true that it has implication on natural resources.